Let's cover some enzymes. So you've got your oxidoreductases, which are based off of redox reactions. So you've got the term Oilrig. The oil stands for oxidation is loss, whilst rig means reduction is gain, and this is regarding electrons. So if it's oxidized, it can either gain an oxygen molecule or lose a hydrogen molecule, while if it's reduced, it can either gain a hydrogen molecule or lose an oxygen molecule. Next up, you've got transferases. These catalyse the transfer of a chemical group between a molecule. So, for example, you've got aminotransferase. And as you probably guessed, this is when an amino group is transferred from one molecule to another. You've got hydrolases, which, as you probably guessed by the name, involve the use of water. So these catalyse the division or splitting of large molecules into smaller molecules by the addition of a H2O molecule. So for example, you've got lactase, which breaks lactose into glucose and galactose by adding a water molecule to it. They can also act in a reverse, so there's a condensation reaction where if you remove water from your glucose and your galactose, then you'll reform lactose. It's not a water molecule from each, it will form a water molecule between the two. So like you'd remove a hydroxyl group off one and a H group off the other, and that'll form water, and then you get your lactose. Similarly to this, you've got lases, which catalyse the breaking of bonds but without water. So for example, you've got your pyruvate decarboxylase. This causes pyruvic acid to convert to ethanol and carbon dioxide because it breaks the carbon-carbon bond, so a carbon can be released. And you may have heard of isomers in chemistry. This is a similar concept. So you've got isomerases, which cause the change of a molecule's organisation. So for example, you've got phosphoglucose isomerase. This changes your glucose 6-phosphate into fructose 6-phosphate. Basically the same thing, just a bit messed up. Last but not least, you've got ligases, which catalyse bond formation between two compounds or molecules. So for example, with DNA ligase, when you have two DNA nucleotides, it forms a phosphodiester bond between the phosphate on the 5 carbon and the hydroxyl of the 3 carbon of two adjacent nucleotides.